Taylor, CEO and co-founder of Precision Nanosystems from Vancouver, talking about meeting the challenges of microfluidics, enabling nanomedicine development from basic research to the clinic. Sorry, I mixed it up, yeah. So next talk is Andreas Fisch from Novartis, Pharmaceutical Development Unit, talking about ZentriMix, a novel screening tool for liposomal formulations in early development.
Yeah, ladies and gentlemen, I'm very happy to be here, invited at CLINAM. Peter was advertising mixed micelles, and I would like to advertise a new technology for preparing liposomes, especially suitable for early phase development. It's called dual asymmetric centrifugation. Okay, yeah. Okay. As you all know, liposomes are the most mature parental nanomedicine, and if you look on the products which are on the market and those in clinical development, you see that most of them are loaded with generic drugs, which are in general available in very big quantities. But now, if you want to develop new liposomal formulations loaded with new chemical entities, you face the problem that the amount of drug you have available at this phase is very limited. So we are talking more about milligram scales rather than gram scales. So what we need here is not only a fast, a simple and reproducible method for production of liposomes, but also a method which is working in very low scale. And here we have some of the conventional methods for preparing of liposomes. To start with the method, the reference method may be the film hydration method invented by Bengham. And there are much more methods how you can produce liposomes, and this list is certainly not complete. I would like to focus your attention on this dual asymmetric centrifugation. And as the name is already implying, it has something to do with centrifugation. So you need a centrifuge, but a very specific kind of centrifuge. It's not only turning in one dimension, but in two dimensions, and with this exerting a lot of shear stress inside the vials. In addition, you add glass beads or zirconium beads inside the vials, so you even increase the shear stress inside the vials. This enables you to work with extremely high lipid loads. So we are talking about 40 to 50% of lipids in this initial mixture. So how is it? And with this, of course, you can achieve rather high encapsulation yields. So how is it working? So you first of all put your lipids, your phospholipids, your cholesterol inside the vials. And a very important point here is that you should have homogeneous mixtures. You can buy them from your suppliers. Novartis, we are doing them by our own. We are just to have more flexibility. So we are dissolving the lipids in butanol and lyophilize them in the vials itself. So then we add a certain amount of beads and the drug solution, and we start the first round of centrifugation. So what we obtain then is what you see on the right-hand side, a very, very viscous system. These are already formulated liposomes, but in such a high concentration that it's extremely viscous and you can do nothing with it. So you're obliged to apply a second round of centrifugation after adding a certain amount of buffer. And then you are in a viscosity range that you can do the classical purification, so remove all the drug not encapsulated inside the aqueous core of the liposomes, for example, dialysis. So here you see that you can play around with the process parameters, and by this you can tune the size of those liposomes. For example, if you look at the red field, if you increase the volume of drug solution, you can bring the particle size down to about 100 nanometer, but then you are apparently reaching a certain limit, so even if you give more buffer, you will not be able to bring it further down. In case you want to have large particles, you can just reduce the amount of beads or the shear stress in terms of rotation speed. And then you come to particles maybe in the size of maximum 600 nanometers. A very interesting point here, or a question, is will the liposomes you have prepared during the first round of centrifugation stay intact and keep their cargo when performing this second step of centrifugation, where you only want to disperse those liposomes? And this question can rather easily assess in just taking different amounts, different volumes of buffer during the second step of centrifugation. In case you will not harm, destroy, or partially destroy the liposomes during the second step, encapsulation efficiency should be the same for all those formulations. But apparently in our case, this is not really the case. If you look at the lower right figure, you see that the encapsulation efficiency is going down with the amount of buffer you're giving, meaning the conditions we have selected here are a bit too stringent. So by reducing the rotations, the RPMs, you might be able then to preserve the liposomes generated during the first round of centrifugation. So when we are talking about encapsulation efficiency, we should also talk about the size, because encapsulation efficiency will drop with when the size of the liposomes is getting smaller, because the inner volume of the liposomes will reduce dramatically with the third potency of the radius. So it comes to a massive shrinking of the inner volume of the liposomes. I tried to modulate this on the right-hand side. I gave, if you start with liposomes with 150 nanometers and bring the size down, the inner surface or inner volume will be reduced dramatically, red curve. On the other hand, if the number of liposomes will increase in your wire, but to a lesser extent. So at the end, you will end up with lower efficiency, with lower size of the liposomes, and this matches very well the reality. So very quickly to the advantage, you are working small scale, high encapsulation efficiency, you need a limited amount of drug substance, you can prepare up to 40 formulations in parallel, well suited for screening. The size can be tuned, it's easy to operate, you get reproducible results, you do not need organic solvents. An additional benefit is that you have a closed system. It's very good for highly active, working with highly active substances to limit exposure. And you can handle this process in an aseptic way to get low endotoxin material for animal testing. The inconveniences are you need homogeneous mixtures, but it is easy to do. The size is only can only controlled in a limited range, so very large ones and very small ones are not so easy to obtain with this technology. Not suitable for shear stress sensitive drugs like proteins. The temperature control is only indirectly, but it can be done with the temperature of the chamber. And of course, the system is a screening tool, is not intended for full scale manufacturing. So I hope that I have convinced you that this technology is fast, simple, robust, and it gives high encapsulation efficiency and with tunable sizes, and is therefore very well suited for the development of liposomes embedded with new chemical entities. The size and the encapsulation efficiency can be controlled and fine-tuned by the process parameters, but the nature of the lipids and the drug substance has to be taken into consideration as well. And here are people involved in our technology platform, Nanomedicine. And with this, I would like to thank you for your attention, and maybe I have some time to take some questions.
Thanks a lot, Andreas. We have time for 2 questions. Please.
Very nice talk. I had a quick question about your dialysis step. So you mentioned that this is a screening tool and it's not necessarily something that you're going to scale up as it is. Mm-hmm. But have you thought about thinking what type of replacement you could use for, I mean, you'd probably not use dialysis if you were to scale it up. So how could you purify your liposomes?
In the screening phase or in the scaled-up phase? Yeah. Yeah, you can do ultrafiltration like Stelian pointed it out. Membrane-based tangential flow filtration would be the most appropriate way to do it.
Other questions?
Hi, thanks for the great talk. I wanted to ask you, so we're working with many groups that need much smaller liposomes. Have you tried a sub-100 nanometer liposomal size with this technique?
We are trying, and sometimes we are getting, but it's not so easy. So 100 is so the natural threshold you can get easily. Otherwise, you have to really work and try to optimize the system to squeeze it. So it's not easy. Easy is from 100 to 600.
One more question? Okay, thanks a lot. There was one. Yeah.
Mm-hmm.
Yeah, thanks. Very interesting stuff. So one question with the NCEs, are you finding that they're mostly hydrophobic or hydrophilic? And how does that impact the screening that you want to do?
Yeah, parenteral drugs tend to be rather lipophilic, so a high percentage of lipophilic drugs. And of course, those could also be integrated in such a system, but then you have an extremely high encapsulation efficiency because you would integrate in the lipid bilayer. So the difficult part is then the smaller fraction maybe of the new chemical entities which are hydrophilic, and for those one, the system is even better suited because then you get really higher encapsulation yields than with other technologies.
Okay. Any more questions? If not,